right, I'm excited to introduce our next speaker and last speaker.
Or advance with the arrows with this.
Sorry, I need some direction here.
That is okay.
These to advance, or you can use the arrows again.
Fine, thank you.
You ready?
Ready, go?
All right.
Okay.
Carl, we'll introduce Carl and let him go.
So, as you probably saw, the title of my talk, I think, was Exploring Stress with Dr. Lamont. And I think, as we've all heard, in fact, Lamont is the cure. And certainly in my experience, that's true, both professionally and certainly also personally in terms of helping one through periods of stress. So thank you for that. And well, you already saw it, but I had a found picture of Dr. Lamont practicing her yoga. I love this picture, so I had to use it. Okay. So we were involved in a USDA grant that was Lamont, Max Rothschild was on it, Chris Ashwell, and Mike Persia. I believe we were all sort of co-PIs on that grant. And the objective of that was, well, the title of it was Adapting Chicken Production to Climate Change Through Breeding. And I had to go back and just look at productivity of that. So we had a total of, I think, 28 publications out of that work. I think I lost count, but we had 39 separate authors on that work. Apparently, it's almost 10% of your cited publications, 1,250 citations as of the 5th. And then at the bottom there, I think number 2 is Angelica's paper. So those are the top 3 in terms of number of citations. So I would argue that that was a very productive and certainly for me, extraordinarily wonderful collaboration. So thank you. And I was just gonna go over just a little bit of the data that came out of that, and I think more importantly, perhaps in the context of heat stress, where the field might go in terms of trying to offset that ongoing problem. So this is just a summary of what goes wrong when almost any animal experiences heat stress. And probably the fundamental cue there is protein misfolding, along with the nervous system detecting that change in heat. And then you have a whole series of molecular responses to that. As you can see, it's pretty complicated and pretty interwoven. And at the cellular level, you have a whole series of responses that can occur. And probably the most significant that's right there is the initiation of oxidative stress. And a lot of the sequelae that come from heat stress come from that in particular. And then finally, at the organismal level, you have these potential problems that arise. And one of the, I think, most immediate that is a real problem is the whole problem of leaky gut and the escaping of endotoxins into the blood supply. And we've heard about that earlier today. But you have all these responses that occur. The other thing that's probably really important is circulatory rerouting, where the blood is rerouted to the skin to dump heat. But one of the major problems that occurs then is you lose blood supply to your gut, which in turn leads to leaky gut. So I think that's the connection there. And this is just sort of a summary of how the body itself responds to heat stress. You have, is this a laser pointer? No. You have a response that mainly gets initiated in the hypothalamus and the pituitary. And you have a neurotransmission response that affects a whole variety of organs, largely, of course, to reduce heat output. And also at the bird level, you have a reduction in feed intake to also reduce heat output. And then You also have the neural control that diverts blood to the skin, which then in turn leads to intestinal vasoconstriction, which then in turn leads to loss of tight junction integrity, which then in turn leads to release of endotoxins, which then leads basically to an inflammatory response and all hell breaking loose. At another level there, you have a pretty severe decrease in immune function, which of course has its own issues. You have reduction in reproduction function, increase in glucose, increase in two other things that I can't see because they're hidden. So this, pardon me? Yeah. So this was the fundamental. What am I doing wrong? Come on.
No, just give it a second.
Oh, give it a second. Patience, something I didn't learn from Sue. She tried her best. So I think this was one of the earliest papers that came out of this work. And again, this may be, I'm not sure if it's Angelica or Mara, I'm not sure. But using GWAS studies to map over about 120 loci responsive to heat stress. And this was in the broiler by Fahmy, advanced in a cross line. And one of the ones that came out of that I've always wished we could have gone back and attacked was a hit on, I think it's chromosome 1, that where 15% of the breast weight, 15% of the variance in breast weight as a function of heat stress mapped to this particular position. And we haven't had the opportunity to go back and actually identify what that is yet. But it certainly conveys, as we've seen earlier with all of Dr. Lamont's input to these kinds of studies, how important these studies really are. The other thing that came out of this work, I think what happened was Max happened to walk through the office when we were trying to figure out how to get to Africa. And I think Sue volunteered him to get us there. And so we wanted to do that, of course, because of the climate in Africa. And another thing that came out of this work was we were also able to go to Brazil and collect samples there. And you can see here the variety of phenotypes in the backyard phlox, naked neck. And then in the middle there, that's just the picture inside of one of the backyard phlox, one of the homeowners, and you can see a whole variety of chickens in there. So you have the ability to look at the genomics of a wide variety of birds that one would argue, since they're really just let loose to mate, that they are under natural selection. So we were very curious about that. And these were some of the runs of homozygosity, some of the regions apparently under selection that came out of this. And they basically encompass pretty much everything that the fundamentals of heat stress response, apoptosis, regulation of that, calcium activation of protein kinases seems to play a role in that. Then, of course, you have basically all the stress proteins, the chaperones in there with environment, oxidative stress. And then in this particular region, since we're equatorial, selection for, I think, is largely response to UV radiation, which a different kind of radiation from heat, but I suspect that's what's going on there. And the other thing we were able to do, as I mentioned, and again, I think Max was responsible for getting all these samples, was looking at SNP panels in birds from Brazil, Sri Lanka, and Egypt. And one of the things that came out of this that surprised me or mystified me a little bit first was basically the birds from Sri Lanka are up there in green. All the ones that are blue are from Brazil. And then at the bottom, those are the Egyptian lines. And apparently, the Brazilian birds show a great deal of similarity to birds from Sri Lanka, and I admit I was a bit mystified by that. until I discovered that, in fact, they were both Portuguese colonies. It's, hence probably the connection between those two types of lines through Portugal. This is essentially a PCA plot where the green, red, blue circle there, sort of in the middle, is close to the black. And so the black indicate the different populations from Brazil that were tested. And then the green, red, blue are birds from Sri Lanka. And then over there on the right, you have the birds that were sampled from Egypt. And one of the things that, of course, one would love to be able to do is, again, exploit these samples further now that we can actually sequence things. These are the selection signatures that came out when we looked at the birds in Sri Lanka. And I would argue that these are not heat stress. They're all involved, basically, it seems to be an innate immunity. So presumably disease selection in that population. And if I remember, very similar responses in the Brazilian birds, and then the Egyptian birds certainly showed examples of heat stress. This was another study that came out of this work, and comparing birds from various locations in Africa with birds from various locations in largely Northern Europe. And big surprise, the population segregate. Up there in the upper right, you have African birds. And there we suspect the continent is largely a proxy for heat. And the lower left there, you have Northern European birds, which apparently, big surprise, are in a very different selection. And these are some of the genes that were identified as proximal to regions of selection. And the ones all in red are ones that also respond to heat stress by increasing their expression level. Having said that, my suspicion is, I think it's a reasonable hypothesis to think that these particular genes are actually responsible for the difference in thermotolerance between these populations. And that perhaps these would be good ones to go back, figure out what the allelic differences are, and think about using those to try and increase thermal tolerance in chickens as climate changes. So this is just the overarching view of the kinds of processes that are under, I believe, are under selection with heat stress. Big one up there on the top is cell signaling. And then I think another vitally important one is the immune system. And you also have, of course, a tremendous suppression of the cell cycle under heat stress. You have a suppression of the vesicular transport system and the cytoskeletal system. You have a pretty great increase in protein folding and chaperones available. Actually, a pretty remarkable impact on lipid metabolism. And finally, you also have an impact on genes involved in circadian rhythm. So this sort of cluster of biological properties is kind of a summation of both the GWAS studies and the studies that we were able to do in Africa and Brazil and Northern Europe. And this is just another display of that. The bulk of them, as you can tell, are largely stress responsive. You also have cell cycle being impacted there, central nervous system being impacted. Presumably that's impact on the nervous system in response to heat stress. So pretty broad variety of systems that are impacted by heat stress itself. And the other thing you can do is apply your transcriptome data and your metabolomic data that we were able to get from these samples also to investigate how different pathways are affected by heat stress. So here we have in red either the genes or the metabolites that were changed under heat stress. In both of these, in these cases, the red were increased in heat stress. So we have an increase in gluconeogenesis in the liver under heat stress, big surprise, the liver is providing glucose for the rest of the system to try to keep the system alive. And very similar pathways can be displayed, one of which actually also drives home the importance of lipid synthesis by the liver under heat stress, presumably to keep organs like the heart going. And this is sort of a summation. So blue are processes that are downregulated, red upregulated under heat stress. I'm trying to be patient. Inhibition of cell cycle, immunity is repressed, beta oxidation is dampened down, glutathione production is increased, pentose phosphate pathway is increased, amino acid levels are reduced except for cysteine, which is the only one that isn't reduced, and presumably that's to keep glutathione production going. And finally, lipid synthesis is elevated under heat stress in the liver. So all of this work is from the liver. So you can see how resources are being shifted, basically, of course, to keep the liver providing to other organs what they need to stay alive, and also things like glutathione to keep itself going. The other thing I just wanted to point out is when you look at heat stress response over longer periods, you get a very dramatically different response the longer the birds are exposed to heat stress. So in this particular model that we use throughout the work, after 21 days, the birds were exposed to another 3 weeks of cyclic heat stress. They were exposed to heat stress for 8 hours, then it went back down to normal temperatures in the evening. So if you look at this particular gene is HSP, heat shock protein E1, under control conditions, you hit the wrong button. You have one level there pretty low. After 4 hours of heat stress, you get a significant increase in expression. But after 4 hours of heat stress, when the birds have been heat stressed for 3 weeks, you get almost a control, if not control levels of heat stress. And so you have a lot of shifting in terms of the response of various genes to heat stress as a function of how long they're exposed to heat stress. And then that's going backwards, we'll try going forwards. So this is just sort of a total count. So as you can see, in the red is heat stress, the blue is the control. Genes that are, what's the right word, downregulated, basically genes that are upregulated in control. So after 2 hours, a significant heat stress response in terms of the number of genes that go up. But after 4 hours, you're already shifting the heat stress response down fairly significantly, I would argue. And after 1 day of heat stress, you see another down in terms of both the heat stress responsive genes and the genes that are elevated in the control birds, until by 3 weeks, it's all pretty much skewed towards elevated expression of the genes responsive to heat stress in the control birds. So this is something I think going forward we have to keep in mind in terms of how to improve heat stress resilience in terms of populations. So response is not uniform, and I think this is probably important in terms of our long-term thinking. That is that the response changes over time as a function of how long the birds are exposed to heat stress. Of course, as part of the acute response, you have upregulation of chaperones and downregulation of these various functions. So now it's just some of the things that I would propose or would be important to do. Of course, one of the ways to help birds survive heat stress is by thermal conditioning of the embryo. So if you thermal condition the embryo, the most common scheme is between E10 and E18, embryonic day E10, embryonic day E18. You raise the temperature in the incubator to 39.5 degrees centigrade, then you bring it back down and let them hatch. Presumably there are epigenetic changes that triggers, that in turn leads to heat tolerance in the adult bird, the post-hatch bird. So this is certainly probably turning out to be one way in which we can improve. But the other thing that I think we're going to have to do is genome editing. And this is just the pathway scheme in which you do that. You grow up primordial germ cells, which you grow out of the 2-day-old embryonic aorta. You then genome edit the PGCs, expand them. You can then microinject them back into the aorta, and you do enough birds, you get a gene-edited chicken. So that's the fundamental process. And these are genes based on, well, the top 3, those are genes that in other organisms that have been shown if you increase their copy number, the resulting organisms are far more heat tolerant. So these are ones that just by increasing their copy number might improve heat resilience in chickens. And the rest of them are ones I admit I just thought of as candidates based on how many different organisms, these particular genes, they respond to heat stress. And also the fact that I'm pretty sure they all map to GWAS peaks. And so I think those would be pretty good targets to think about. And in particular, of course, I think what we need to do is get the chicken alleles that confer resilience so that you're not turning a chicken gene into a human gene or that kind of thing, but rather through gene editing, you're just swapping alleles. Okay, so I think we need to, as have been mentioned, continue evaluation of feed and water additives. I think probiotics, at least some experience we've done, look very promising. Understanding of how the biotics work, which seems to be a bit of a mystery, even I think still. Associated genetic variation with heat tolerance. So what alleles are going to allow for better heat tolerance? One possibility is simply increasing copy number of genes through genome editing, adding extra copies. And the nice thing about this, I believe, is one could, well, by definition, if you want to make a chicken, you have to gene edit primordial germ cells. And one thing you can do, of course, is test the primordial germ cells in vitro to see if those cells are a bit more tolerant to heat stress. So you have a nice experimental system, I think, right there. And then the other thing that one I think would be worth doing is simply taking primordial germ cells and increasing the temperature in the incubator until you reach a point where cells survive that died when you started the experiment. So simply in vitro selecting for heat tolerance and then seeing if the chickens that come out of that are heat tolerant. So, no questions at the moment. So first, of course, thank you, Sue. I mean, look at this room. And we all owe you in many, many ways. And I'm sure most of us would say both professionally and personally. I certainly know that working with you had a big impact on my career. It may well be your fault I got tenure, which I appreciate. I'm not sure the rest of the faculty did, but thank you. And if you don't mind, Sue, if I weren't so manly, I'd probably weep a little bit. Would you all stand up and just thank her? And I think I finished on time. Sue. So a while ago, Sue yelled at me for always going over. So from now on, I try not to. One learns one's lesson from Sue.
It is 3:59 by my watch, so not only did you start a little bit behind.
But just barely.
Yeah, you finished ahead of time, although you just walked away for no questions.
Oh.
You have a question?
Oh, maybe the one.
I don't know if that was a slide move, but.
Yeah, thank you very much. I highly doubt that the chickens have yet come to roost. I think there is still a lot in the tank. So my question is about this heat stress work. I know you've given that feature list, but I'm wondering what is foreseeable nearest, because I know the world is worried about temperature rises, but there are particular places which are really hot spots. And so the fear is the chicken maybe is out of the way soon. So I don't know what are the things we can grab on. I've seen your features list, but I think it is far, especially for some vulnerable areas.
Yeah, I mean, those, to my mind, were things that might impact the high production industry, right? And I think if I understand your question, right, how do we take care of, if you will, the small farm holder, those birds? Yes. Well, I think that an understanding of a lot of the stresses that impact birds is coming from and is going to continue to come from those backyard flocks, since they've been under natural selection. And then the real challenge, of course, is you would like to improve the birds that are used in backyard flocks. And in one sense, I think that's very possible. Although you saw the variety of birds that were in, that individual's house. So I think the problem would be to sort of standardize the birds that are used in the backyard flocks. And then the other challenge is you have to be aware of the cultural preferences. Different places may put bigger importance on feather coloring or something like that from other places. So obviously it's going to be a little more complicated, but I think it's incumbent upon us to do so, to try and help those backyard flocks, given how important they are to survival and economy. I know that's a hand-waving response, but to be honest, that's the best I can give you right now.
Okay, I will take that. If you don't mind, half a question.
Uh-oh.
So the other one is yes, half a question. So there was this presentation about the Turkey Center, Evaluation Center. I actually wanted to ask, but I will now ask, is there any work on heat stress in turkeys? I think there was no mention of that, but maybe the one to you, because now they are of the stage. Is there another bird other than the chicken? Is there a work that has compared different birds?
Well,
Hopefully that you can.
First, Kent's right next to you. Certainly their group is working heavily on Turkey. I've seen several publications on heat stress in Turkey. And one year we made the foolish attempt to try and combine our efforts and get funding from NIFA to jointly look at heat response in chicken and turkey, but oh boy, they didn't like that one at all. But yeah, that was very soundly rejected. But yes, I'm actually, I have to admit, if I were thinking about doing this when I was 30 years younger, 30 years I'll be dead. 30 years younger, I would probably give serious consideration to quail, because of the rapid turnover and the fact that in many countries they are an important part of food resources.
Thank you.
Thank you.
Questions?